to do a brief review. We're looking at the citric acid cycle. I like to draw it like this, in the sense that there's that, what I usually call step 0, but it's where we have to convert pyruvate into what's the actual compound that pyruvate gets converted into before it can enter into the citric acid cycle. Acetyl-CoA. And so it has to be converted to acetyl-CoA. And that's why it's directly not a part of the circle, but it's really important. It has lots and lots of the cofactors and everything that's involved. Remember, it converts, whoops, then I'm going to run out of room. But ultimately, you go from an NAD+, and every time there's an NAD+, you have to have a proton, by the way, and to make NADH, which what class or classes of enzymes can do that? Dehydrogenase, and this one was called pyruvate dehydrogenase complex. That's my abbreviation. All right. And the other cofactors was the fact that there is an FAD, and there was a, TPP, thiamine pyrophosphate, and as well as lipoate, or lipoic acid.
And those are separate from the complex, or those are part of the complex?
Those are, well, in the instance of the TPP and the lipoate, those are prosthetic groups that get added on, like functional groups that get added on to part of the complex through a lysine, through 2 different lysines. And now the FAD is actually something that's different, but it doesn't leave as an FADH2. Remember, it automatically gets regenerated back to FAD in order to make the NADH. And the reason why I want to keep pointing that out is because what we'll find out when we do the electron transport chain is if you can make an NADH, ultimately it will make 2.5 ATPs, mole per molar, okay? Whereas an FAD can only make 1.5. And this will make sense whenever we go over the electron transport chain, because right now, there are reasons why we use FAD, FADH2. There's redundancy that's available, and hopefully whenever we do the electron transport chain, it'll now this makes so much sense. Life is sweet, kumbaya. All right, and then we covered the first step, just real briefly. The fact that was to make citrate, and that was citrate synthase. Here, let me go on. And citrate synthase, one thing I forgot to point out is note that it is irreversible. And so just like glycolysis, you need to know the steps that are like the checkpoints. One checkpoint, in a sense, is step 0, but it's why you have to be really careful the way the question may be worded, because it's like a gateway to help control the citric acid cycle, but it's not a part of the citric acid cycle directly. Well, this is step 1, it's citrate synthase, it's allosteric, and almost every time that when we're talking about allosteric, we're talking about something that's important here. And it should make sense, both NADH and ATP inhibit it, because that means there's lots of energy. And then the newbie that we hadn't seen before was succinyl-CoA. And we'll find out succinyl-CoA is one of the other products made during the citric acid cycle. We just aren't there yet. And then we did step 2. In step 2, you took that citrate. Oh, and other things to point out was this is the reason why it's called the citric acid cycle. It's also called the tricarboxylic acid cycle because there's 1, 2, 3 tricarboxylic acids on there. But citrate becomes isocitrate. It does have an intermediate, what was the name of that intermediate? Connotate. This one is different since it uses iron as a cofactor. It does that whole Ferris wheel. One other little side note that's always this interesting little tidbit is the fact that this was achiral and does make it chiral, okay, which we don't see it a lot, especially in vitro. We just can't do that, but our body utilizes that. Right? And the enzyme that does this is called a condensation. For whatever reason, it's named after the intermediate, which does not leave. That was one other thing I forgot to point out again on the pyruvate dehydrogenase complex. It never leaves. The in between steps, it just gets passed off. It's just a little assembly line. So it's important that you don't have the condensate just leaving per se. Okay. That was just, we went over one iron sulfur center. This is just one example. Iron sulfur centers a lot of times are also involved in electron transport or electron shuttling and things like that. And we'll see that a lot during the electron transport chain. All right, now we're to step 3. Step 3 is to go to isocitrate. Come on now. Wake up. It goes from isocitrate. Nope, that's not what I wanted to do. This is isocitrate to make what's called alpha ketoglutarate. A lot of times, I'll tell you right now, I'm going to abbreviate it as alpha Kg, just because we're going to see this is important. This is another intermediate that's involved in multiple pathways. Okay, and so that's why if you see me doing alpha Kg, just remember that's alpha ketoglutarate. I'll try to say it at least once every time I go to use it. Just what's one observation that we can make about this? NAD plus to NADH, and so what class or classes of enzymes can do this? This is a dehydrogenase. Okay, what else? What's another observation that we can make? Right, and so it's a decarboxylation reaction, but this one is a dehydrogenase. What else? What's just another thing that maybe it pops out at you, or maybe it doesn't, but that you should take note of? It is an alpha keto group, so that's important. Actually, we're going to come back to that one. So make note so don't forget. Technically it's called an alpha keto acid, right? That's another thing, like real subtle but very important here. Pardon? That is. We're gonna get to that one. Everyone's like, we have isocitrate, we have NADH, we talked about oxaloacetate, which is just an intermediate. It's decarboxylated. This is to show you the mechanism. Okay. It makes alpha decarboxylate. So what's the one thing that we have not circled right there? It's irreversible. It's one of the checkpoints. Like literally, we're doing every single thing. The other box, there's just its name. And so we know it's a dehydrogenase, and so if you're going to name this one, what would we name it? You could think that. What's the other option? This is isocitrate dehydrogenase. Yes. This one, the name makes perfect sense. It's dehydrogenating. my use of the word, but no, it's doing isocitrate. Okay. Now we're going to cover back to the alpha keto acid. So, let me just draw it out bigger. Make sure I don't miss anything. CH2. This. So this is alpha ketoglutarate. It's an alpha keto acid. Where have we seen an alpha keto acid before? What was the other example that we? Alanine. And so when we did alanine, what was the part that we changed to an NH3 or to the amine? Right, it's a carbonyl. So now if we just draw the same thing, CH2, carbon. But I'm going to go ahead and a different color put in. I'm going to draw it this way, but it could also be an NH2. It just wouldn't be positively charged. What did we just make? And by the way, this is reversible. We can go back and forth. What did we just make? Someone said it. This is glutamate or glutamic acid. So alpha ketoglutarate and glutamate are related. Does anyone remember what was the enzyme that converted alanine into pyruvate and vice versa? What are we adding to the alpha ketoglutarate? Amine. And so what would you suppose, what kind of enzymes can add that? It's aminotransferases. And so, yeah, we're going to see that this enzyme here is called an aminotransferase. Okay, and it's reversible. We're going to see it over and over and over again, just like we do with alanine and pyruvate. Okay, this is one of the checkpoints. There we go. So, and once again, it should make sense. Like it's going to be inhibited by ATP and NADH because we have lots of energy around, so we don't need to make any more. It's going to be activated by ADP and NAD plus because that means that we need energy, and so it's going to turn this enzyme on. Okay. Now we're to step 4. So we have alpha-ketoglutarate. Okay. And we're making succinyl-CoA. So, which we've seen, succinyl-CoA, of course, is one of the parts that can go back and inhibit, if it starts to accumulate, it inhibits citric synthase. Pardon?
Is there no enzyme involved?
There is an enzyme. There's just, because I took that from the old book, and so it doesn't have a space for the name. So I just want you to think about, you can think about where we've seen those same types of cofactors and like what's going on, all that kind of stuff. I'm looking for this. I thought I had another figure to me up, and I must not have. In the world. Okay. I thought that I put in a different figure there, but I didn't. My bad. Okay. So what does this enzyme look like? What's going on? It's a complex, exactly. We have NAD plus becoming NADH. We're removing a CO2 group. We have, what is this? That's coenzyme A. So it's being attached this time. So we're making acetyl-CoA, we're making succinyl-CoA. We've got magnesium, of course, TPP, lipoic acid, and FAD. And so where did we see those same kind of, it was pyruvate dehydrogenase complex, and so what one do you suppose this one is called? This is alpha-ketoglutarate dehydrogenase complex. Here we go. Now, I want to point something out here. Even though this one is showing it as being reversible, the important thing to note here is this is also the third checkpoint. Okay? Of the direct, not including step 0, we had step 1, citrate synthase, we have isocitrate dehydrogenase, and we have alpha-ketoglutarate dehydrogenase complex. Just
all irreversible so far?
No. Steps 1, 3, and this one in vivo is going to favor going this direction. Go towards the succinyl-CoA.
So step 2 is not irreversible?
Step 2 is reversal. Step 0, step 1, step 3, and step 4. Okay, those are the checkpoints. So 1, 3, and 4, the citrate synthase, isocitrate dehydrogenase, and the alpha-ketoglutarate dehydrogenase complex are all the checkpoints within the actual circle, and step 0 is a checkpoint outside the circle, like an entryway or gateway.
So step 2 is irreversible, but not the checkpoint.
Right. Okay. Okay, let's see. Is that all I was going to say about this one? Let's sidetrack. Right. So one thing to also note is so far we haven't made any energy directly from citric acid cycle. Remember from glycolysis we made 2 ATPs off of the pyruvate kinase enzyme, but so far we haven't made anything directly. We made NADHs, which can go on in the electron transport chain, but there's nothing that's made it directly. So now we're coming up to step 5. Step 5 is the very first time where we're going to directly make some type of energy. Here we have succinyl-CoA, which becomes succinate. It utilizes a GDP to make a GTP. You get your coenzyme A back, so that way you can go on to do, whatever acyl transfers, or it can be involved in like acetyl-CoA formation or succinyl-CoA formation. What class or classes of enzymes could this possibly be? Okay, kinase. All right, quite possibly a kinase. What else could it be? It's not a pyrophosphatase. So what else have we seen that utilizes or could possibly a class of enzymes that could do like ATP, GTP? We have kinases and we have, someone said it, synthetases. So this one is a synthetase because for whatever reason they decided to name it for the reverse, even though it's essentially irreversible because you're making ATP. I mean, ultimately, but they did. This one's called succinyl-CoA synthetase. I know it's one of the ones that people sometimes forget. And so I think in the newer book they've changed it to where they made it look better. Here, I'm going to make this a little smaller. And the reason why is this is the way that we've been showing the citric acid cycle. And right here or so is where what happens is we make a GDP turn into a GTP. That doesn't directly make ATP, but what happens here is an ADP gets converted into an ATP by a side reaction. And it's the same thing that we've seen before. We actually saw this one before earlier. We talked about it with UTP, remember? And when we did the epimerases and the UDP transferase, glycosyl transferases, the way that you got the UTP back, this is the same enzyme. It's that nucleoside diphosphate, which one way to abbreviate that is NDP, nucleoside diphosphate kinase. Because all it does is it transfers a phosphate between two different types of nucleotides. Almost always, I can't think of any exceptions off the top of my head. ATP is usually one of them. Either ATP is donating it to make the UTP in the earlier example, or here the GTP is donating its phosphate to make ATP. Okay, so this is the succinyl-CoA synthetase enzyme. Then we have the next two. So we take succinate, and I'm actually going to cover them both on the slide before I go on. We take succinate, we're turning it into fumarate, and FAD becomes an FADH2. This is one of the only times that we've seen where the FADH2 then goes on and leaves. And what we'll find out in chapter 20 is that that's going to go make 1.5 ATPs. Okay. So what class or classes of enzymes utilize FAD and FADH2? The dehydrogenases. So because that's what this is. This looks different because always before when we did redox reactions, we turned like alcohols into ketones or like an aldehyde into carboxylic acid. But remember, one of the other shortcut methods was if anytime that you increase the number of carbon to hydrogen bonds, you're reducing it. And if you notice, we're increasing the number here, and so therefore this is another type of oxidation reduction reaction. So this is a dehydrogenase. So if you're going to name it, what could you name this one? This one, and I just don't have enough room, but this is succinate dehydrogenase. Okay. There's also something else that's really special to point out, very important about this one. This enzyme here is the crossroads between it and the electron transport chain. So if you think way back to one figures that I drew early on when we started talking about metabolic pathways, I tried to show how the various metabolic pathways are all interconnected. And this is one that directly connects citric acid cycle to the electron transport chain. And so if I was going to try to draw the, which I don't know that I'm going to be able to do it justice. Oh. Okay, so that's supposed to be the citric acid cycle. And then here's the electron transport chain. That's where they intersect, is right here at succinate dehydrogenase. It's called complex 2 in the electron transport chain. Okay. So that's where they intersect. And another important thing to point out is this is all in the, it's supposed to be the mitochondrial inner membrane. And so the electron transport chain occurs there, and this is the only part of the electron transport chain that kind of sticks out into the mitochondrial matrix. Because the citric acid cycle takes place in the mitochondrial matrix, but there's a portion of complex 2 that sticks out, and that's where the FADH2 is made. Okay. And then we have fumarate. Fumarate becomes malate. So water gets added, and that's what malate looks like. And what this enzyme is one that's got a special name. This one's fumarase. And I'm just going to click on the next one so that way you can see it's spelled and it looks prettier than my handwriting. This is fumarase. We're almost back full circle. We've actually seen malate before. Where have we seen malate? It's part of, what was that metabolic? Inadvertently part of another metabolic pathway. Gluconeogenesis. I mean, it doesn't actually take part in it directly, but remember, it was helped with the shuttling. It was called the pyruvate malate shuttle, where it could, because what malate can get through the membrane, but what could not? OAA. And what did we have to do? When we started step 1, it was acetyl-CoA, and acetyl-CoA reacted with, what did it react with to make citrate? OAA. And so that's the 8th step. So malate is another way that we kind of intermingle, so to speak, with gluconeogenesis. But this is the 8th step. You have to go from malate to oxaloacetate. We've actually already seen this. This is not a new enzyme. What's one thing that we can, what's one observation about it? It's a dehydrogenase. And so if we were going to name it, what would we name it? It's malate or malate dehydrogenase.
Because I think on the other one there was like systolic.
There's cytosolic and there's mitochondrial versions. Okay, for this one, I mean, if you wrote malate dehydrogenase, I'd give it to you. But for when it's a part of the citric acid cycle, technically it's going to be the mitochondrial version.
Right.
When it's a part of gluconeogenesis, it's both, right? Because part of it takes place in because this one's even though it looks like it's irreversible, it's reversible because I copied that from a previous book and I can't change it. And so it's reversible, and whenever it's in the mitochondria, it goes backwards from here from OA to malate, and then it goes out to the cytosol and becomes malate to OAA again. So therefore it goes back and forth.
So were steps 6 and 7 reversible as well?
Let me see. That's why I thought I had the one figure that I like better. Yes. The truly irreversible steps are, says step 0, 1, 3, and 4. Under physiological conditions, I should say, under normal conditions. Okay. So overall, for every pyruvate, you get 1 GTP, and that 1 GTP gets converted to 1 ATP. So sometimes in some books, they'll tell you, oh, you get 1 ATP, but it's from that nucleoside diphosphate kinase. You get 1 FADH2, you get 4 NADHs if you include the one from step 0. Now, if you don't, then it's only 3. Because remember, that's why we said starting from pyruvate, we're going to get the 4th. But if we say starting from acetyl-CoA, you only get 3. And you get 3 carbon dioxides. And remember, a carbon dioxide is just a carbon that's used up all of its electronic energy. Okay, and so, I mean, it's fully oxidized. You can't make it any more oxidized than that. No, this is just the same thing. This is a summary. All right, so the 3 checkpoints, we've already discussed this, but this is just to, I don't know if I have any, if I put that in, because there are no boxes, because it's just a little summary on there, but that was the fact that we had citrate synthase, we had isocitrate dehydrogenase, which would be quote-unquote step 3, we had alpha-ketoglutarate dehydrogenase complex, okay, and then we have that step 0, as I like to call it, which is pyruvate dehydrogenase. Oops, it should say complex, and it doesn't. My bad. Here's a little cartoon showing all of that, if you prefer cartoons instead. But I really like this one in the sense that it shows you the 3 checkpoints, oh, the 4th if you count that, but the checkpoints, it shows you which ones have the NADs and the FADs in there. And, oh, whoops, it does not show you where the ATP is made. And so if you want to add that in, ATP gets made right here, GDP to GTP. And then, whoops, I'm going to run out of room, but ADP to ATP. Okay, this is important. Whoops, not what I wanted to do. We've actually been saying this all along. This is just specifically putting it out. When we say that whenever there are lots of ATP, then that means there's going to be very little ADP, and so they like to talk about an ATP to ADP ratio. Okay, so it's rusting. Same thing with NADH being higher than NAD plus. And so then the ratio is high. The exact opposite happens whenever you're in active, you're being active. Okay, now is little ATP and high ADP, little NADH and high NAD+. Okay, this is where it gets fun. Fun's been fun all along. Because remember we talked about how the citric acid cycle is involved in both catabolism and anabolism. And what I want to point out is this is where we really get to where you start to see where all these things start coming in together. And we've only seen so far carbohydrates and proteins in a way. Okay? Because what happens is in catabolism, we went through glycolysis to make pyruvate, which converts to acetyl-CoA, and goes right in to make the citric acid cycle intermediates. We've also talked about amino acids, because what amino acid, just a good review, what amino acid gets made into pyruvate? Alanine. So that means you can break down your proteins with proteases, those are the enzymes that break it down, to the amino acids, and then alanine, for example, gets made to pyruvate, which gets made this way. We've also made what amino acid gets made into alpha-ketoglutarate, part 1, glutamate or glutamic acid, and so that's just another example. We'll see that there are others. There's one that you may already be able to figure out if you take your time and you look at all of the intermediates. One of the other intermediates of the citric acid cycle is also an alpha-keto acid that has a direct amino acid counterpart on your own. Well, I mean, actually, I think the next slide goes over it. And so we can see how it gets. Now, we haven't covered fatty acids yet. That's later this semester. But what we'll find out is fatty acids get broken down to make acetyl-CoA, which can go on to make these. So this is how it's involved in catabolism. This is just a little cartoon showing the exact same thing, but it can be a little daunting, but I don't want it to be, okay? Because I want you to be able to rationalize it and be able to see like how hopefully it's starting to make sense. Because it just blows my mind. It's a beautiful thing. There's our mitochondria. Okay. So what happens, this TA, the old enzyme name for aminotransferases were called transaminases. Sounds like a car. But it's an aminotransferase. So that's where every time this says TA, that's an aminotransferase. But we've already shown here, oh, isn't that so sweet? We've already shown here where alanine using aminotransferase gets made to pyruvate. Pyruvate gets shuttled, or it gets transformed into acetyl-CoA. This is the citric acid cycle. We're going to find out how you can have other amino acids and stuff that can be broken down to acetyl-CoA. Acetyl-CoA can go in through, and so on and so forth. We've already talked about glutamate, making alpha-ketoglutarate, but you can just start to see how these all play a part. The one I wanted to point out, and if you draw it out, oxaloacetate is the alpha-keto acid of aspartate. We're also going to find out later on this semester, asparagine can be made directly into aspartate. Just like glutamine can be made directly into aspartate, arginine can be made into aspartate, proline can, and so, I mean not aspartate, to glutamate. And so you'll see the same thing over and over again to where we can break these things down in order to make energy when needed. And on Friday, we'll talk, we've actually almost finished this chapter, where I'll talk a little bit about how it can be used for anabolism, to where you can utilize these intermediates to ship them out to make bigger things as well. So I'll go ahead and I'll